In this segment, I'll talk about the thyroid hormones and regulation of metabolic rate. The thyroid hormones, thyroxine, T4 and T3, are key to balancing metabolic rate, the use of energy, with the availability of glucose and other energy sources. The active form of thyroxine, T3, is moved into cells through a transporter where it binds to its receptor, which is a transcription factor, which moves into the nucleus, binds to DNA, and either increases or decreases messenger RNA production and proteins. The outcome of T3 winds up changing protein production for three major uses. One is that it tends to increase the production of glucose from precursors. Two, it increases the release of free fatty acids from fats, both of which are energy sources for cells. And three, it tends to increase protein synthesis. The thyroid hormone system is regulated by the brain and pituitary. Here's an anterior pituitary, much larger. TRH is released from cells in the hypothalamus to cells in the anterior pituitary that release TSH down to the thyroid glands, a loosely organized gland in the area of the throat, where binding of TSH to receptors results in a second messenger activation that activates co-transport of sodium, which moves in iodine. It also activates the enzymes that make the thyroid hormones T4 and T3. T4 is called T4 because it has 4 iodines, and T3 has 3 iodines. And we'll get back to why both active T3 and inactive T4 are secreted. TRH neurons in the hypothalamus are regulated by either other neurons or by receptors themselves that respond to glucose levels and free fatty acid levels, two major energy sources, and they respond to cold temperatures and other inputs. Cold increases TRH release, it increases metabolism. High glucose and or free fatty acids increase TRH secretion. So when food is abundant, TRH secretion is high and metabolic rate goes up. When individuals are lower on food, either because there's insufficient food or perhaps they're dieting, the low glucose and low free fatty acids decreases TRH secretion. And so unfortunately, a response to dieting is simply to reduce metabolic rate to compensate. So TRH from the hypothalamus stimulates TSH from the anterior pituitary, which stimulates T4 and T3 in the thyroid. T3 increases activity of cells and therefore metabolic rate. There's a feedback pathway of T4 onto TRH so that if T4 and T3 production are very high, we inhibit TRH. So why have both T3 and T4? So T3 is responded to by most cells to increase activity, increase metabolic rate. T4 can be converted to T3. But why do that? Imagine a group of cells that are carrying out normal functions, but a separate group of cells that's being used heavily or repair and recovery from injury. So these cells may need a higher metabolic rate. T3 that's everywhere in the body is moving onto these cells and causing a baseline metabolic rate for the body. But T4 will be moved into cells, converted to T3. And so in cells that need to have higher metabolic rates, they receive both the T3 that every other cell gets, and either produced in their own cells, they get additional T3. we wind up with one group of cells that just has the baseline metabolic rate. We wind up with a second group of cells that has that same baseline, but in addition, they have this additional T3 that's being developed by this T4 to T3 pathway. So by secreting thyroid hormones in both forms, inactive T4 and T3, the body can have a baseline for all cells and increased T3, increased metabolic rate for any tissue that needs to have a higher metabolic rate at the time. The thyroid hormones are not highly soluble in water, and so in the liver, cells secrete binding proteins, T3 and T4 binding proteins, that in the blood will bind to T4 or T3 and carry them to other tissues. So in the blood, there's always some T4 and T3 that gets free of these binding proteins. They have high specificity, but not high affinity to the thyroid hormones. And so they'll be popping off and popping back on. In any given tissue, it's the free T3 and T4 that can get out of capillaries into extracellular fluid, and from there into cells. So these binding proteins in the liver help carry T3 and T4 through the blood. How do you get rid of T4 and T3? For what's called clearance, all hormones have to be cleared and removed, otherwise once secreted, you could never turn off the effect. Again, in cells in the liver, there are cells that have the enzymes to convert T4 to reverse T3, which is an inactive form of T3. It has an iodine in a different location, and convert T3 to T2. These move into capillaries, which now carry T4, T3, reverse T3, and T2. And in the excretory tubules, blood is coming in carrying T4, T3, reverse T3, and T2. Remember that capillaries are leaky, and so is the beginning of a nephron, an excretory tubule. And so T4, T3, reverse T3, and T2 all wind up in the excretory tubule, and they're never recovered, so they're excreted with the urine. So the hormones are cleared in two ways. A, enzymatic degradation, which is common for hormones in general, and also common for hormones, B, urinary excretion.